My name is Jing-Ke Weng. I'm a researcher at the Whitehead Institute. I'm also an associate professor of biology at MIT, and you are in my lab. In this lab, we study plants. Plants have been around for a very, very long time, so the ancestors of plants were aquatic plants in ocean, and around 470 million years ago, they came onto land, and now they are everywhere, and they were very successful, occupying every single corner of the planet, and we use plants in our daily lives. And in this lab, we're really curious about the chemistry of plants. What are the very interesting compounds that could be useful for humans, and how they are manufactured by plants, and how we can harness the science we learn from them to engineer better products that can enhance life. So this is a very typical molecular biology laboratory, and you see a lot of containers with a lot of solutions. The pipettes we use to essentially pipetting very precise volume of liquid into tubes and to do mini reactions. So a very typical workflow for the lab is we get a plant of interest into the lab, and we perform an exercise called sequencing. So nowadays we can use this technology called next-gen sequencing to get massive amount of genetic information of any plant of your wish, and then extract particular genes that encode the enzymes of interest, so then we can study the exact function of that particular enzyme. So once we figure out the function of every individual enzyme, then we can put them together. Hopefully we could assemble a pathway making molecules of interest. Many of the molecules we're interested in are medicinal, so that means although they were produced by plants for their own purposes, humans have discovered particular medicinal properties of them. So in the traditional sense, they'll be extracted from the native plants in a way. If the plants are endangered, and the exercise could be very harmful for the environment, but use the new technology we're developing here, once we know the genes that are responsible for making those specific molecules, then we can reassemble the whole pathway in a different host. A very typical example would be bacteria, E. coli, or the baker's yeast, and then we can use large-scale fermentation to produce exactly the same molecule used to be extracted only from plants. So that's a very new technology and very, very exciting time to study plants. Yeah, some of the molecules plants make a very low abundance, and it is very inconvenient for people to have to extract enough amount that can be used in medicine. Now, with the new technology we're developing, we could really amplify the production of these low abundance molecules in another setting. Another extended benefit of this approach is we could make so-called new-to-nature molecules. Some of those molecules may never exist in plants, but we can add additional catalysts to convert them into new sets of analogs. Maybe some of those analogs are even more potent in terms of being developed into a medicine. The current pharmaceuticals that we can get from pharmacy are usually single molecule treatment. These treatments have limitations. On the other side, the traditional medicine coming from plants have a lot of molecules in them, and we are still at a very, very early stage to trying to figure out what are in these traditional remedies. But by discovering more why and what molecules and why these molecules are present in specific medicinal plants, we may unveil new disease mechanisms. Maybe a set of combinations will be the most optimal for treating complex diseases.
Hi, my name is Colin, and I'm a second-year graduate student in the Weng Lab. So I did my undergraduate training as a biochemistry major, and one of the fascinating things that drew me into this laboratory is the immense amount of chemical diversity that plants have. So plant natural compounds are pretty useful and very bioactive, and they're widely used as therapeutics and also for biomaterials. And one type of research and one research work that I'm currently working on involves halogenated plant natural products. And halogenated plant natural products compared to bacteria are pretty rare in nature. However, they harbor very interesting bioactive properties. And one of the example being acutamine, which is a chloroalkaloid compound, which is tetracyclic in nature, and due to its complex chemical structure, it's very interesting to observe how plants are able to naturally make this chemical compound. And the plants that make this compound are the moonseed plants, which are Menispermum canadense and Cinnamonium acutum. And these moonseed plants are able to make this because plants are able to create this complex biosynthetic pathways, and it involves this natural halogenase that is able to halogenate a pretty complex compound. So these are moonseed plants. These are specifically Menispermum canadense, and these seeds make these moonseed plants that produce the halogenated alkaloid compound that is bioactive. So halogens are pretty important, and they're pretty potent, and they're needed in a medicinal compound that serve as anti-cancer or antibiotics and for therapeutical purposes. So plants are excellent chemists, and they're able to do this with biosynthetic enzymes. There are proteins that are macromolecules in the plants that are able to create these complex chemical compounds. And one of the research work that is being done in our lab is to take these proteins and engineer them to carry out novel biosynthetic reactions to derivatize natural products and create new properties and create new therapeutics that are of biomedical interest.
So my name is Tomáš. I'm a postdoc here in the Weng Lab, and my main project since I started here has been focused on a Polynesian plant called kava. So kava is a herb that people drink, and it has relaxing and soothing properties, and it has been shown in some clinical trials that it can reduce anxiety and depression. It's available commercially. You can buy it in a dried powder form like this. And in Polynesia, people drink it from a traditional bowl. It's called tano, that you can see here. And you drink it from a coconut shell. So we have some coconut shells here, so we can also try the effects of kava ourselves. And so we are interested in the active molecules in this plant that interact with the human brain receptors. I have the molecules right here. So these are the active molecules in kava that interact with the human brain receptors, and they're called Kava Lactones. And we are interested in how the plant makes these molecules. So using modern technologies like sequencing and mass spectrometry, we analyze the genes that you can find in the plants and that are responsible for producing these molecules. And now we are trying to engineer this biosynthetic pathway into a laboratory microorganism like yeast. So the reason why we are interested in Kava Lactones is that it has been shown that Kava Lactones can activate the GABA-A receptors in the human brain, which are the receptors that are often targets of common antidepressant medicine. However, these molecules activate the same receptor but through a different site. So they can regulate the GABA-A receptors through a different site than the traditional medicine that you can buy. So we potentially have access to a completely new way how to modulate this function of the brain. I think there is a huge potential for the future development using the biosynthetic power of plants, because plants have had millions of years to evolve amazing molecules, and we have only explored a tiny fraction of those.
Hi, my name is Matthew Hill. I'm a graduate student in the Weng Lab, and I work with a different system than the rest of the lab works on. I work with actually plant cell culture, which is different than working with entire plants as you might find them in a greenhouse. It's actually just sort of a soup of individual plant cells that we can work with much like we would with a bacteria or a yeast. You don't have to grow them like full plants. You don't have to wait a month before they're ready for experiments. They're continuously ready to go. And I'm working on methods to take some of the methods we currently use in full plants and move them to this system so we can accelerate the pace of our experiments and hopefully the ease with which we conduct experiments. These are the tobacco cells. They're from the stem of a tobacco plant. These are from actually the 70s, developed in Japan as a way of the tobacco industry trying to produce nicotine without actually having to grow tobacco plants. These actually contain no nicotine whatsoever, but they've since become very useful to the plant biology world, the molecular biology world, for studying plant cells in ways that are very difficult with full plants. And they're not green because they're from the stem of the plant, and the name of the cells is BY2, bright yellow 2. And so what we can do with these is actually transform them with various bits of DNA, make them express genes that they would not normally, and then essentially see what the effects of those genes are. What we can do with these cells is use different laboratory methods to express different genes in them. These might be genes that other people in the lab work with if they're trying to express enzymes that are more difficult to express in the full plant or that you can't express in bacteria. Because these are more close to your average green plant, a natural plant that you'd find in the greenhouse or in the wild, we can support different enzymatic activities that are not possible in hosts like bacteria or yeast. So the fact that we're working with a plant system that is more simplified and somewhat easier to work with than an entire plant allows us some ease of access when it comes to this complex biology. The ultimate goal of this project is actually to develop a system that uses plant viruses encoding different genes to actually make enzymes better and more efficient and even make novel products. Nature has created such a diversity of secondary compounds and metabolites that we work on, especially in plants. But right now we're limited to the availability of what nature provides for us. And there's actually a bottleneck of having to go out into the wild and discover these enzymes and these compounds before we can use them. We're hoping to use this system that I'm working on to, instead of exploiting nature, taking the forces of nature into our own hands and actually changing the enzymes to better suit our purposes, to make new products or make the same products more efficiently.
My name is Mike Torrance Spence. I'm a fifth-year postdoc in the Weng Lab here, so I'm doing postdoctoral research. We have broad focuses in this lab, so there's lots of projects that I'm sort of interested in. Most recently, I've been curious about the biosynthetic pathway for making this plant-critical defense hormone called salicylic acid. This is a pretty common and simple small molecule. People are probably most familiar with this as an acne medication. It's used for drying skin and sort of combating acne. It's a super cheap chemical, so we're not interested in synthesizing this for commercial production, but because its role as a plant defense hormone, it signals the plant defense response. And so this is common amongst all plants. And so this has enormous agricultural potential. If you're able to elucidate the pathway, then you can manipulate the pathway, and we can hopefully overexpress salicylic acid or some analogs in various agricultural plants to make them super pathogen resistant. Again, with climate change, there's all sorts of introduced pathogens. Plants are becoming increasingly more sensitive because the natural habitat they're growing in is changing. It's getting hotter. There's new pests. There's possible sea level rise. There's increased salinity. There's increased pollution in the soil and the air. So if you can make these plants more resistant, then this is enormous. There's growing population by, I think some estimates, by 2100, there's supposed to be close to 10 billion, I might be mistaken there, people on the planet. So we need basically more food with less space to feed more people with a changing climate. And so I think we're approaching this from a basic science perspective. We want to know how these plants make this chemical, but it's very applied, I think. And it's coming down to something very simple like what we eat every day. People don't think about this. People probably rarely think about the sources of their food or the difficulty farmers might have in producing it, but this is an emerging problem. So we're hoping to address this. So in this work I'm describing, we've figured out how plants broadly make salicylic acid, this simple chemical. This chemical itself does not fight off pathogens, but it signals internally in the plant that it's under attack, and then the plant is able to bolster its own defense responses. In this case, we found that in this pathway, there's a specific enzyme, which is what we're looking at here, which is this 3D structural model solved from experimental data, where unlike most plants, there's a certain group of plants that only contain this enzyme. And this enzyme accelerates the production of salicylic acid. And so this is in sort of the mustard family. So this is in some agricultural plants already. But basically, if we're able to take this enzyme and put it in other agricultural plants that do not contain this gene and this corresponding enzyme, then we think it can accelerate the production of this defense hormone and then further bolster defense responses, which is fantastic for hopefully enhancing crop production. So in this case, we would hoping to be engineering these genetically modified organisms that produce this chemical that bolster the native responses, and with the plants, utilizing its evolutionary response to pathogens which have evolved and are very sophisticated to produce thousands of defense proteins and defense compounds, it'll be less necessary, hopefully, to be using lots of pesticides and chemicals and fertilizers, which are polluting the environment, ending up in the ocean, causing oxygen loss in the ocean with excess fertilizers and so on. So we hope to sort of make cleaner agriculture with genetically modified organisms that contain this enzyme, amongst others.
Hi, my name is Sophia Xu. I'm a fourth-year PhD candidate at MIT Biology, and I work in the lab of Dr. Jing-Ke Weng here at the Whitehead Institute. I have been working on the kudzu vine and trying to understand how the flowers of this vine can alleviate hangover. So this vine, the flowers of the kudzu vine are used in traditional Chinese medicine to alleviate hangovers. And my project in the last several years has been trying to identify a compound or class of compounds that is responsible for this effect in humans. I am also working on the biochemistry, the biosynthesis of sporopollenin. So sporopollenin is a tough, durable, chemically inert biopolymer that coats the outside of pollen. And earlier this year, our lab solved the chemical structure of sporopollenin. So now we're trying to understand the biosynthesis. So how are some of these enzymes that have been previously identified, how are they involved in building up this polymer, and how are they adding like functional groups? And once we understand what these enzymes are doing in the plant, or in these plants, because all land plants make pollen, we make sporopollenin, we can start to understand how can we co-opt these enzymes to make a polymer that we can apply in other aspects of human life. So you can imagine, like, say, your bike is getting really rusty in the winter. So can we build some kind of a plant-derived polymer that we can just spray onto your bike to protect it from rust in the winter? So applications such as these. Another thing that we're interested in this lab is understanding how climate change affects these plant natural products. So we know from the literature that climate change impacts these plant natural products, both in their biosynthesis and in their chemical structure. And some of these mechanisms are known, and others are entirely unknown to us. So we're interested in learning how, or a better understanding how, these mechanisms or climate change is affecting